Hi, today's video is going to be all about proteins, and we are going to talk about what proteins are and what they're made of, and all the different types of functions that proteins have. So the title of our notes today is going to be proteins. And proteins are very important, and all living organisms, they make proteins. So if you eat any living organism, you're eating a lot of protein in that. Because most of the time when you think about protein, you usually think about meat like chicken or steak. But all living organisms have protein, including plants. But before we get into what proteins look like, I want to show you a picture. I'm going to show you a picture of a tool, and I want you to think about what the function of this tool is. Okay, so here is a picture of a tool. And just looking at that, what do you think it does? I'll give you a minute to think. Maybe you think that it's a tool where you're going to kill zombies. There's going to be a zombie apocalypse, and you're going to have this tool, and you're going to have two of them, and you're going to kill all the zombies as they attack you. Maybe, right? But looking at this tool, you can see that there is a handle. So obviously there's a place for you to grab with your hand, and then there's a hook on the other end, and that obviously hooks into something. So based on the structure of this tool, you can guess what this tool is for. And I'm going to show you what the tool is. So this is what we call a hay hook. And so here is a farmer, and she has two of them. And so you grab two of them, and then you can stick them into the hay, and now you can lift these big hay bales, and move them in different places. So the point of me showing you that is that all things, especially protein and all biomolecules, they have a particular shape, and the shape is directly related to the function. And so proteins have a large range of different functions, where before, when we talked about carbohydrates and lipids, their function is for storage of energy. But for proteins, there isn't one general function. Each protein does a different job. And so sometimes proteins are known as molecular machines. So proteins are molecular machines and have a wide range of functions. So I'm going to show you a few examples of some proteins, and just this is not an exhaustive list. There are thousands of different proteins. So I'm just going to show you a few just to give you an idea of what a protein looks like and maybe what it does. So here is a protein, and maybe you recognize it. So this protein is called an antibody. Okay, and so an antibody is a protein that is in charge of finding and labeling pathogens in your body. So antibodies are part of your immune system, and antibodies are a protein, and they circulate through your lymphatic system and your circulatory system. And what they do is they locate things that should not be in there, and they bind to them, and they label them for destruction. Okay, so an antibody's shape has two arms. Okay, so if you see at the top here, these are like two arms with hands on them. And the two arms are in charge of grabbing onto pathogens, has two arms that are able to bind to pathogens. Okay, and this binding labels a pathogen for destruction. So there are other cells in your body. For example, there are T cells, and there are killer cells, and there are macrophages, and there are other cells that if they see something that is covered in antibodies, they know they need to attack that pathogen. Okay, so that is one function, and you can see the structure has two arms so that it can do its job. So I'm going to give you a couple more examples. So here is an example of a protein. And this protein is called collagen. Okay, so this protein is called collagen. And as you can see, it is just like a rope. It's got this rope structure. And so what collagen does is it's a protein that binds bone to bone, okay? Or it can bind bone to muscle. So it's like a rope because it attaches two things to each other. And so again, you can see the relationship between what it looks like and what it does. So collagen is in charge of binding two parts together like a rope. Okay, and I'm just going to show you one more example. Let's see here. I'm going to show you my favorite protein. Okay, so this is my favorite protein of all time. It is called a kinesin. Hang on. Kinesin. And the protein is actually the colored part. So it's the blue and the red part. And what it is the protein that moves, and its job. So you can see that it has like these 2 feet, and these are like the 2 legs. And it actually walks through your cell on its 2 feet and its 2 legs. And what it does is it carries cargo. So kinesin has 2 legs and feet, and it walks inside the cell like a little man and carries different cargo. Okay. And so this like little man is inside your cell doing all this work. And this is the cargo here. It's like something big, something that has to go from one side of the cell to another side of the cell. And it gets like on this kinesin and it travels through the cell and it literally walks. And I will show you a video of this when we get to, I will show you a snip of video when in my video about cells. Okay. So those are some examples of protein. And what I just want to focus on is just that these proteins look very different. There's a very, very wide range of the way proteins look. And that is because they have a wide range of functions. And so to get into the nitty gritty of what a protein is all proteins are made of monomers. Proteins are made of monomers. And those monomers are called amino acids. Okay, so there are amino acids, and there are 20 different amino acids. Okay, so each amino acid has a basic structure, chemical structure, and I'm going to draw that. So in the middle of each amino acid, and there are 20 different ones, there is a central carbon, okay, because all these biomolecules have carbon. So I'm going to put a C there for carbon. It's in the middle of an amino acid. Each amino acid also has attached to it a hydrogen, okay? And then it also has attached to it what we call a carboxyl group. And we're going to write COOH. That's what a carboxyl group looks like. And then attached to it also we have an amino group. And that's where the word amino acid comes from. Okay, and so an amino group is a nitrogen with 2 hydrogens attached. Okay, and then all carbons have to be bound to 4 things. And so the bottom side group is what we call an R group. Okay, I'm going to do a different color. And so an R group is a variable group. I'm going to write that down. Variable group. And it varies. It's different for each amino acid. And there are 20 different amino acids. So each amino acid has a different variable group. So there are 20 different variable groups. Okay, and I'm going to show you a table that shows you all 20 different amino acids. Okay, so here's a table that shows you all the amino acids. And because, people love to categorize things, it just helps us understand and put things in different categories. And so these amino acids are based on some properties that they each have. So there are 4 groups. There's a nonpolar group, a polar group, a positively charged group, and a negatively charged group. And so for each amino acid, in the nonpolar group, we have glycine, alanine, valine, cytosine, proline, leucine, isoleucine, methionine, tryptophan, and phenylalanine. And all of these, you can see, have the central carbon. There's a carboxyl group, and there's a hydrogen, and there's an amino group for each one. And then in the yellow box is the variable group. Glycine being the most simplest, the variable group being just an H, a hydrogen. But you can see that some of these variable groups are really large. But all of these amino acids are nonpolar. Then we have the polar group, which includes serine, threonine, tyrosine, asparagine, and glutamine. Then we have 3 positively charged amino acids, which is lysine, arginine, histidine. And then we have 2 negatively charged amino acids, that's aspartic acid and glutamic acid. And so these amino acids have these properties, and it is very important that they have these properties, and I'll tell you why in a second. So a protein is made up of a string of amino acids. So each one amino acid is going to bind to another amino acid, and that's going to bind to another amino acid, and that's going to bind to another amino acid. And each protein has anywhere from 100 amino acids to thousands of amino acids. So some proteins are really small with only 100 amino acids, and some are very, very large with thousands of amino acids. And so when you have 2 amino acids that bind together, I'm going to move my picture here, and I'm going to show you how they bind together. Okay. So here we have 1 amino acid, and I'm going to add another amino acid here. So it has a central carbon, and then we have a carboxyl group, and then we have a hydrogen. Then we have an amino group with 2 hydrogens, and then we have a variable group. Okay, and that's just different depending on what amino acid it is. Okay, so what ends up happening is when this amino acid binds to this amino acid, this OH comes off and this H comes off, okay? And now we're going to have H2O be removed. And this is called a dehydration synthesis. So what ends up happening is we have the central carbon of one amino acid bound to the carboxyl group, which has just one oxygen now, and that carbon is bound to the nitrogen of the next amino acid, and that's bound to just one hydrogen because it lost one. And then here is the central carbon of the next amino acid. So here is the carboxyl group, and this is a hydrogen. Excuse me. This is a hydrogen. And then here is the variable. I'm going to do that in blue. And then here's the amino group, NH2. Okay, and I'm going to put the variable groups here so you know the orientation. And so right here, how this carbon binds to this nitrogen, this is called a peptide bond. Oops, hang on. Okay, so this bond right here is called a peptide bond. Oops, bond. Okay, and so this is one amino group attached to an amino acid, attached to another amino acid, held together by a peptide bond. Okay, and when you have a long string of amino acids, that is called a polypeptide. And a polypeptide is another way of just saying a protein. Okay, so I'm going to write another way of saying protein is a polypeptide. Okay, polypeptide means you have lots of amino acids strung together, and that's what a protein is. Okay, and each amino acid is held together by a peptide bond. So when you have a protein, they have all these different shapes. And so as a scientist, you need to be able to describe each protein. And the way that we describe the shape of proteins is through these different characteristics. Okay, so different levels of description. So I'm going to write this down. Let's do, what color is this? Purple? Okay, that's just black. describe a protein, we describe it at 4 different levels of description. Okay, that sounds kind of confusing, right? But let me explain. So whenever you're describing a protein, because they are very complex proteins, very complex structures, the way you do it is you go through these different levels. So the first level is called the primary structure. Okay, so there's 4 different ones. So the first one is called primary structure. Describes the sequence of amino acids. Okay, and so another way that we write primary structure is like this. You write it like a 1 with a little degree sign. That means primary structure. Okay, and an example would be something like you would write methionine, then that's attached to a leucine, that's attached to a glycine, that's attached to a tyrosine, that's attached to another methionine, and so on. Okay, on and on and on. So the primary structure is just listing the order of the amino acids that are in the protein. Okay, so the sequence of amino acids. The second thing is called the secondary structure. So let me number these. That might be easier. So that's the primary structure. And then the second level is called the secondary structure. And the secondary structure describes 2 common shapes that you find in proteins. So it describes 2 common shapes that emerge when there is a polypeptide chain. Okay, so let me explain. When you have this long string of amino acids, so let me just draw that here. So let's say this is an amino acid and that's bound to another amino acid and this one's to another and on and on and on. Okay, so this is what a protein is made of, different amino acids. And I showed you in this table here that some of them have a positive charge. So I'm going to put a plus here. And then some of them have a negative charge. Okay, so let's say this is histidine here. And this negative charge one is aspartic acid. So this negative is going to be attracted to this positive. And then it's going to cause this string of amino acids to fold like this. See? So let's say this is the positive one and this is the negative one. Those 2 are attracted to each other. So it bends. And because of all these properties that the amino acids have, polar, nonpolar, positive, negative, when you have this long string of amino acids, because each amino acid has a property, it just folds up into this shape automatically. And there are 2 shapes that are super common. And that's what is described in the secondary structure. So those shapes are alpha helix and beta sheet. Okay, and these are just super common shapes that automatically form when you have a polypeptide chain, and so they get their own category of description. And so let me show you the double helix, and then also I want to just write here that secondary structure is written like this. You do a 2, and then you do a little degree sign. But let me show you what the alpha helix and beta sheet look like. So an alpha helix is just this helical shape. So it just looks like this, okay? And then the beta sheet is like when you make a fan out of paper. So you know how when you get really hot and you're like in school and you don't have a fan on you, but you need to fan yourself because you're so sweaty. You fold up a paper back and forth, and then you slightly open it up so that you can just fan yourself. Okay, so this drawing is not the best, but you get the picture, right? It's like this zigzag fan. That's called a beta sheet. So this is called an alpha helix, and this is called a beta sheet, okay? Alpha helix, and this is a beta sheet. Okay, and so let me show you a picture out of your textbook to show you what these look like. Secondary structure. And so here's a picture out of your textbook, just to give you a better idea. So here's the alpha helix. And so these amino acids just automatically form this shape based on the properties of the amino acids. Okay. And then here are beta sheets. There are 3 here. So you can see they just zigzag back and forth, like in and out of the plane of the screen. Okay. So that is, that describes the secondary structure. Then we move on to the tertiary structure. Tertiary structure. And the tertiary structure is a little bit more messy. Okay, when you see a protein, like when you saw that, the antibody, it just looked like a blob, right? And a lot of proteins just look like a blob. So you have these alpha helices and beta sheets that automatically form, but in between the alpha helices and beta sheets, you just have all these like, blobbish formations. So the tertiary structure describes the in-between structure. I'm going to say in-between structure between the alpha helices and beta sheets. So you could say something like there's one beta sheet and then there's like 2 bends and then a beta sheet attached to another beta sheet attached to another beta sheet and then 5 bends like that. So it's not straightforward, and it's just the in-between connections between the beta sheets and the alpha helices. Okay, and then the last structure is called the quaternary structure. Quaternary structure. I knew I was going to misspell that. So quaternary is spelled like this. Quaternary. And the quaternary structure describes the number of polypeptide chains in a protein. It's not always just one. So you saw that there was a string of amino acids. Some proteins need more than one polypeptide chain that get folded up differently and that they come together to make a functional protein. So quaternary structure describes the number of polypeptide chains. Okay, it's not always only one. Some proteins need more than one piece to be functional. Okay, and I'm going to show you an example of that. Okay, and the example protein that I have is hemoglobin. That's another protein. And so hemoglobin is a protein that you make in your cells. And hemoglobin is inside your red blood cells. Okay, so let me write this down. Hemoglobin is inside your red blood cells. Is inside red blood cells. And there are 4 different polypeptide chains. 4 polypeptide chains. Each polypeptide chain is colored here. So there's a gray, a red, and then we have 2 blues. Okay, but each one, there's 4. And each group, each polypeptide chain has a little pocket. And in that pocket is an iron. Okay, so I'm going to say in each chain, there is a pocket with iron. Okay, Fe, that's iron. Okay, and iron binds with oxygen or it binds with carbon dioxide. So I'm going to say iron binds with either oxygen or carbon dioxide, depending on if the blood is going to the lungs or leaving the lungs. And so the function of hemoglobin is to bind to oxygen so that the oxygen can go to all the cells in your body and pick up carbon dioxide as waste. And then it goes back to your lungs and you exhale the waste. So you inhale the oxygen, it goes to a hemoglobin, and each hemoglobin can hold 4. And then the hemoglobin goes to your cell, drops off the oxygen, picks up the carbon dioxide, comes back to the lungs over and over and over. And so that is the function of a hemoglobin protein. Okay, so the last thing that I want to show you is I have a list of proteins here. It's a table that I took from the book, And I just want to show you some of the variation in proteins. So this is showing you a list of different proteins here on the left. It's telling you the number of amino acids. So it says number of residues, which means amino acids. And so this little one, cytochrome C, only has 104 amino acids. And that's probably the smallest protein. And then we have titin, which is found in humans, which is the largest known protein. And it has 26,000 different amino acids. And then you can see here that some of the proteins have more than one polypeptide chain. So the cytochrome C protein, which we will learn about later in the class, only has one polypeptide chain. And then this glutamine synthase has 12 different polypeptide chains. So it just varies. Oh, and here's hemoglobin, which we just saw. Hemoglobin has 574 amino acids, and there are 4 polypeptide chains, just like we just saw. Okay, well, I hope this was good, and have a great day, and I will see you next time.